from undergraduate to the MS program. The requirement for that is that you need to have a GPA of 3.3, and that will be active for the fall semester. If you have more questions regarding the fast track program, please contact your graduate advisor, Dr. Hu, or myself, and I can give you more information. I also remind you there are scholarships. This is a time for scholarships. There are plentiful scholarships available, and the deadline is approaching soon, end of February, and so you may have gotten notification from your advisors about the scholarships ranging from the College of Science and other private sponsors. So please take the opportunity and do apply for a scholarship. That's in particular for undergrad students. If you want to participate on field camp, there's field camp scholarships. So make sure that you apply on time. Okay, then it's my pleasure to introduce a speaker for today. Isla Castañeda from the University of UMass Amherst. She is working in the area of biogeochemistry and molecular sciences. Isla Castañeda, Dr. Castañeda graduated, is currently associate professor and graduated from the MS, made an MS from UC Boulder, then PhD at the University of Minnesota, worked also at the Large Lake Observatory there. If you think Texas is cold now, if you ever has been in the wintertime in Duluth, Minnesota, you know what's really cold. Then she continued her PhD, so she made a PhD at University of Minnesota and then continued her postdoctoral research at the Royal Netherlands Institute and then became faculty. She has her research is supported by the IODP and the NSF Paleoclimate program. This is also a special lecture supported by IEODP, the International Ocean Drilling Program. And Dr. Castañeda had also several important high-ranking papers ranging from the Journal of Geology, many other high-ranking journals, and also Proceeding of National Academy of Sciences. That said, we are looking forward to have her. I'm delighted to have her as a speaker here in our seminar series, and we leave the room for Dr. Castañeda.
Great. Well, thank you for that introduction. It's great to be here today virtually. And just from my end, it's a little weird because in front of me, I cannot see anyone. I can just see my slides. So that's a little bit of a strange experience, but I'm just going to go for it and hope it goes out okay. So the way my talk is set up is I've been asked to say a little bit about ocean drilling. So that will be the first part of my talk. For like the first 5 minutes, I'll tell you a little bit about the ocean drilling program here in the US. And then I have quite a bit of background. So I just want to make sure everyone's on the same page and can understand my results. So I'm going to give a lot of background, and then the results will come in the latter part of the talk. Okay, so ocean drilling here in the US began in the 1950s and late 50s with Project MOHO, which ran from 1958 to 1966. And you may have heard of some of these acronyms that I'm going to be telling you about below here. So, for example, the Deep Sea Drilling Program, or DSDP, ran from 1968 to 1983. The first decade of that was funded by the US only, and then it became a real international effort. DSDP was followed by ODP, the Ocean Drilling Program, which ran from 1985 to 2003. And from 2003 to 2013, we then had IODP, so that's the Integrated Ocean Drilling Program. And since 2013, the acronym has stayed the same, so IODP, but we're now the International Ocean Discovery Program. And one of the reasons for this change is just that there's a lot more research. We go out to the ocean and we drill ocean cores, but the research is, beyond that of just like drilling. So the current IODP program is funded through December 2024, and at the moment, we're not sure what lies ahead. So at the moment, the US and a bunch of different countries are involved with discussions trying to figure out the future of ocean drilling. One of the main challenges we have to overcome at the moment is that the drilling vessel we've been using for a really long time, it's called the JOIDES Resolution, is an aging ship at this point. It's sort of at the end of its lifespan. It cannot probably keep going for much longer. And so one of the major challenges at the moment is trying to find funding to replace the drill ship. So we will wait and see what lies ahead for the future of ocean drilling. Ocean drilling is a really international effort, and that's one of the things I have enjoyed with being involved with ocean drilling. And if you sail on one of these expeditions, you really are going to be working with an international team of scientists, and you will be remaining collaborators with them for a long time in the future as you work up these results. So where do we have ocean drill cores from? Well, a lot of places in the ocean. And so in this map, you can see all these little dots are places where one of the different expeditions has gone, and they're color-coded by which leg it was. So like the DSDP ones are in green, some of the more recent ones are in hot pink, and this map is actually like a little bit out of date. There's a couple expeditions that have gone that are not yet appearing on this map. But overall, you can see that a lot of our seafloor we do have drill cores from, but there are some places where we have some big gaps. So for example, here in the Southern Ocean, there's lots of places in the Pacific that could benefit from having some more coverage, and currently the coverage is pretty sparse up in the Arctic Ocean. So there's certainly a need for us to go in and get other cores from other parts of the ocean. For those of you who like Google Earth, and kind of a fun tool that I use a lot in my teaching is to, if you can go to this website here, and you can download a Google Earth KMZ file that has the location of each one of these ocean drilling cores that's ever been collected on it. In Google Earth, you can then click on an individual site. It will bring you a link to all the scientific data at that site. It will tell you like how old the core is, what's been done in terms of research, and you can even download the data. So this can be a really fun teaching tool. So if you like Google Earth, I recommend going there and downloading this tool. If you'd like to participate in ocean drilling, there are options to do so. For example, you can sail as a shipboard scientist or a technician. You can serve on an IODP Science Advisory Panel. You can serve on the US Science Advisory Committee. Anyone who has funding and like a laboratory to do research in can request samples and data for research. Graduate students can apply to sail. A number of my own graduate students have sailed on different IODP expeditions to get the samples for their PhD research. IODP also offers Slanger Ocean Drilling Fellowships, so this will support graduate student research related to IODP cores. And both undergraduates and graduates can assist in post-cruise research. And so there's some websites at the bottom if you want more information about ocean drilling, or you can sign up for an electronic email list. So this map shows sites where I have worked. So these are all different ocean drilling cores. And so I've either worked on these sites myself or through collaborators or where my own students have worked. And in 2015, I sailed on IODP Expedition 356. This went to the Northwest Australia Shelf, and each expedition is about 2 months long. So that one I was out for 61 days. But today I'm going to talk about a much older core. So this is ODP Site 660. This was collected in 1986. And one thing with ocean drilling is that you saw that map that had all those dots from around the globe. On each one of these expeditions, multiple cores are collected, and a lot of times maybe only 1 or 2 of them are studied in real detail. So sitting in different repositories around the world are all these ocean sediments that have not been studied in a lot of detail. And I think there's a real opportunity to come back to some of these sites using newer sort of techniques that we have now developed in the last decade or two, and to apply these to some of these older ocean cores to gain insights into things like Earth's past climate. So my own research involves studying Earth's past climate, so the field of paleoclimate. And I often get asked, why do you want to study past climate? And the reason is that Earth is really old. So Earth is about 4.5 billion years old. But our period of instrumental or direct measurement, so this is like someone going out and measuring the temperature with a thermometer or someone measuring the amount of rainfall, those instrumental records are really, really brief. In the best cases, maybe they'll extend back to about 1800 AD. If we can then go to historical records, so this might be like a written record of someone saying there was a drought this year or it was a really, really cold winter in a certain year. And using historical records, we can extend our knowledge of past climate back to about 1000 AD. If we want to go further back in time, we can turn to tree rings as a climate archive. So tree ring records, we can go back in time about 10,000 years. And we have ice cores at both poles. And presently, our oldest ice core comes from Antarctica, and it expands back 800,000 years. Now, 800,000 years is a long period of time, and these ice cores have been very valuable for telling us about how Earth's climate has varied in the past. But still, 800,000 years is relatively short when we think about the fact that our planet is 4.5 billion years old. So if we want to study Earth's climate, before 800,000 years ago, we need to turn to other types of archives. And this can include things like ocean sediments, lake sediments, and also rocks. So today I'm going to be talking about some of the records we can gain from studying ocean sediments. And the reason we want to do this is we want to extend our instrument base and historical records further back in time to get a better idea about how climate varies naturally and over the different time scales that Earth's climate can vary. And one of my own research interests is on studying past intervals in Earth's history when it was a little bit warmer than today. And the reason for doing this is that this can give us insights into like the processes and mechanisms that operate on a warmer world, and it can give us some ideas as to where we are headed in the future. Okay, so with ocean drilling, we go out with our ship, and from the ship, a bunch of pipe is laid down, and it will go through the whole water column. In some cases, this might be 6 kilometers worth of water. The ship can drill in really deep water, and then it may drill a couple kilometers down into the sediments. So we can get this sedimentary record. And ocean sediments are a great climate archive because over time, different organisms living in the water column will fall down to the bottom, they'll accumulate. And so over time, we get sediments accumulating with the oldest sediments at the bottom and the youngest at the top. And here is an example of some different photographs of the cores. And when you just look at these core photos, you can see that there's some visible changes in the coloring here. So you can almost see there's some like layering in this, and you can see it's undergoing some color changes here. From here, it's more like a black color, then it's a greenish color and a brown color. And so once we get the core up, usually the first thing that is done is we date the core to get a good age model. And then we can start to look at it. We can look at what microfossils are in it. We can look at its geochemistry. So that basically these color changes you're seeing here are reflecting something different that's happening in terms of the climate or the environment that these sediments were deposited in. So normally, one thing that is commonly reconstructed from ocean cores are records of sea surface temperature. What I'm going to talk to you about today is how we can use ocean cores to actually study past continental climate. So one of the places where I've worked and where I did my PhD research was on Lake Malawi, which is located here in southeastern Africa. Now, when you have a place on the continent where we have a really large and deep lake, these large deep lakes are also really good climate archives. So like, for example, underneath Lake Tanganyika, there's something like 5 to 10 kilometers of sediment. So this is an amazing climate archive. Now, if you wanted to go to a place like North Africa, so North Africa today is occupied by the Sahara Desert. It's one of the most arid conditions on the place on Earth. But in the past, this area has been much wetter. So there's been times in the past where we've actually had trees and lakes and up here in the Northern African continent. Now, in places like North Africa, where the climate has gotten wet and then it's dried out and it's gotten wet, and these cycles have occurred numerous times. What this means is that there's not a lot of places that we can go to on the North African continent and get a really nice continuous sedimentary record, which is what we want if we're trying to study past climate change. So what we're going to do today is we're going to go to the ocean sediments offshore Northwest Africa. And our core site is located here, shown by this star, and it's situated underneath this large plume of dust that's blowing off the African continent. And as the winds blow, it picks up this dust, and the dust contains in it different types of molecules that were either present in the soil or from the plants living on the land, and they carry them offshore. And then when the dust settles on the ocean surface, eventually it makes its way down into the ocean sediments. So using this approach, we can go to ocean cores located underneath dust plumes adjacent to continents. And then from these ocean cores, we can study past continental climate change, which is one of the things I'm the most interested in. The set of tools I'm going to use today are called biomarkers. These are organic compounds preserved in geological environments, so things like rock, sediment, petroleum, or coal, that we can trace back to a specific source, organism, or process. And an example of a process would be something like photosynthesis or biomass burning. So if we go out to our ocean core, and let's say we're located near the shore, and we get some sediments from this site, what we're going to see in those sediments, we're going to see organic compounds that are produced by the plants living on the land. We're going to see a whole bunch of different compounds that are produced in the water column, so by different types of algae, microbes, archaea, and bacteria that live throughout the water column. We may see some contributions from organisms that are living in the soil and the sediment. And if we're looking at recent sediments, we may also see some anthropogenic compounds. So there's compounds that are produced by burning fossil fuels that we could see in our sediments as well if we're looking at recent sediments. So one of the first things we can do when we start working at a new site is just to see what is there in terms of the organic compounds. And all these little structures on here are representing different types of organic molecules that we might find in the sediments. And so first of all, just by seeing what's there, it can give us some idea about the past environment in which the sediment was deposited. Now, for some of these molecules, they make small adjustments. They might change like the number of double bonds, the number of rings, or the number of methyl branches in response to the temperature at which they grew at. And so a lot of organic compounds can be used to reconstruct past temperature. In some cases, we can look at how these molecules have been modified as they get buried, and they may undergo some exposure to heat and pressure. So in some cases, we can learn about the depositional history of the sediments by looking at the organic compounds. And what I'm going to focus on today is information we can gain by looking at the stable isotopic composition of an individual compound. So this is a field called compound-specific stable isotope analysis, or CSIA. And I'll explain a little bit later why exactly we're going to do that. So overall, these organic compounds that we call biomarkers are really useful tools that can give us information about what the past climates and environments on Earth were like. Now you might be asking, why not just use DNA? DNA is indeed the ultimate biomarker because DNA can give us information about what genus and species was living there in the past. The issue with DNA is that it does not preserve as well as these lipids or molecules. So I will say there's been a lot of recent advances in sedimentary DNA. We can now use this technique further back in time than previously, but still compared to lipids, lipids can be preserved for millions or even billions of years. Now, the difference between using lipids or our biomarkers here versus DNA is that we don't get a distinction down to the genus and species, but using biomarkers, we can really broadly, it's really easy to distinguish the three domains of life. So here we have the eukarya, the bacteria, and the archaea. And for example, within a particular domain, we can distinguish different groups. So for example, there's biomarkers for sponges, flowering plants, conifers, dinoflagellates, and so they don't get down to the genus and species level, but they can tell us about broad groups of organisms. So how do we go about measuring biomarkers? Well, we first start with freeze-dried sediments. And if any of you have ever made like a cup of coffee or a cup of tea, you have done an organic geochemical extraction. So if you're going to make a cup of espresso, instead of starting with sediment, you'd start with some espresso grounds, and you would put that into an espresso maker. This machine here is called an accelerated solvent extractor, and it's basically like a $65,000 version of an espresso maker. You put your sediment into this little cell here. The machine then will pump organic solvent through it. So instead of using water, as you would to make a cup of coffee or tea, here we use organic solvents like methanol and dichloromethane. And then we get out an extract. So this would be equivalent to like your cup of coffee. We call this a total lipid extract because it will contain all the solvent extractable lipids that are in your sediment sample. They are often colored kind of yellow or green or brown, and these are either pigments or degradation products of pigments, mainly chlorophyll. Now, at this point, we could run the total lipid extract on one of our instruments, but it may have thousands of compounds in it. So normally what we do is we do a further step of column chromatography to separate out compounds based on their polarity. So depending on the goals of the project, we might separate out between 2 and 10 different fractions. So in this example, we have 2 fractions, an apolar one, which is clear, and then the polar one, which has all those pigments in it and is a greenish color. And then the last step of this is the sample gets transferred to a small 2 milliliter vial. These fit in the auto samplers of our instruments. And then we'll run the sample on different types of instruments to analyze a different fraction of the organic matter and to identify and quantify the compounds. So one of the main tools we use is gas chromatography, or GC. And a GC is just like a fancy oven. So initially we'll inject a small amount of sample, maybe like 1 microliter, onto the GC column. And a GC column is just a very long, thin capillary column. It's like a long, thin coil. We use 60-meter columns. And so initially, the inside of this column is really, really small. It has an internal diameter that's smaller than like one of your hairs. And so initially all the material sticks onto that column, and then the GC oven will heat up. So in the course of an hour, we'll go from 70 degrees to 320 degrees, and at the same time an inert carrier gas flows through the column. So the combination of this temperature gradient and the inert gas flowing through this starts to sweep different compounds off of the column, and as they travel through this length of the 60 meters, they start to separate out. So the first things to arrive at the detector are the light, small compounds, and the larger, heavier ones come out at the end of the run. And so this is an example of what we might see from the output from a gas chromatograph from an apolar fraction. So on the x-axis, we have time here. And on the y-axis is just a relative abundance. And what we're seeing here, this is just a class of compounds, straight-chained hydrocarbons called N-alkanes. And what we're seeing here, you can just see the number of carbons here are numbered. So this one has 25 carbon atoms in the chain. And what you're seeing here, these sort of regularly spaced compounds coming out, is just adding 1 more carbon atom to that chain length. So this is just an example of what we get out from a GC. We can then take the same sample and put it through some different instruments, like we'll put it through a GC coupled to a mass spectrometer where the compound is ionized and fragmented. This is what we use to identify our compounds. And we can also run it through an isotope ratio monitoring mass spectrometer to get isotopic measurements on individual compounds. So today I'm going to focus on a group of compounds called plant waxes. You may have heard these called leaf waxes, but plant wax is a better term because they're produced by all parts of the plants, including like leaves, stems, and roots. So what they are, they're just a waxy coating on the plants. They help the plants to retain moisture. And when it rains or when the wind blows, these plant waxes get eroded, and eventually they can get deposited in things like lakes, soils, and ocean sediments. And these are one great class of biomarkers that are popular to study, both because they're fairly easy to measure and they're well preserved on geological timescales. Now, if we look at plant waxes, here are 2 different examples. So what we're seeing here are plant waxes extracted from a banana leaf and one from a tropical grass. And the carbon number is indicated at the bottom. So like this banana leaf has some of the alkanes that have 25 carbon atoms. It's producing a lot of the one that has 29 carbon atoms. And then the longest chain length it makes here is 31 carbon atoms. If we compare this to our tropical grass, in this example, the tropical grass just produces 2 of these compounds with 31 and 33 carbon atoms. So in general, we see that on the African continent, a lot of times trees will have a dominant chain length of 27 or 29 carbon atoms, whereas the grasses, the 31 or 33 carbon atoms will dominate the chain length. So one technique we can use to look at how vegetation has changed through time is to look at sort of the distributions of these plant waxes. But one limitation here is that, some species that maybe have lived like 3 million years ago, maybe don't live today, and so we don't have a modern analog to extract to see what those distributions were. Another thing we can do with plant waxes, and this is what I'm going to focus on today, is we can measure the isotopic composition of our plant waxes to gain information either about the plant type that existed or about the water cycle. So if we take one of these plant waxes and we measure the carbon isotopic composition of that, so the delta 13C, This can tell us about the type of the photosynthetic pathway that the plant used. So I'll call your attention first to this diagram here. And this is, so plants use one of two main types of photosynthetic pathways. The first is called the C3 pathway, or the Calvin-Benson pathway. And this is named such because as the plant is taking CO2 from the atmosphere and is starting to make glucose, in the C3 pathway, the first step that happens produces a 3-carbon molecule. So that's why it's called C3. Most plants on Earth use the C3 pathway. This includes like trees, shrubs, and cool season grasses. Now, some plants, and especially these that are in the tropical part of the African continent, use a different pathway, and this pathway is called C4. The reason for this is that in the C4 pathway, first a 4-carbon molecule is produced. So today in tropical Africa, C4 plants are things like warm season grasses and sedges. And compared to C3 plants, they have a higher water use efficiency, so meaning that they're drought adapted. And they can also thrive in conditions when atmospheric carbon dioxide concentrations were lower than they are today. So like today, we are somewhere around like 415 parts per million by volume CO2. And, the last time you see that in the geological record is about 3.3 million years ago. So when atmospheric carbon dioxide concentrations are lower, you often have these C4 plants can thrive better. Now, the reason we can tell these apart with their isotopes is that C4 plants have a mechanism to concentrate carbon dioxide within their cells, and this causes them to be isotopically enriched in the heavy isotope, so 13 carbon relative to 12 carbon. So if we were to go and measure a single N alkane, if it comes from a C3 plant, it's going to be more negative or more depleted. It might have an average value of about -35 per mil or parts per thousand. Whereas a C4 plant is going to be a little bit isotopically heavier or enriched in the 13 carbon, and it will have a value of about -22 per mil. So we can use this to determine in the past whether we had C3 or C4 plants. And if we had C4 plants dominating over C3, this might point to there being lower atmospheric CO2 conditions, higher temperatures, or arid conditions, as all of these can favor our C4 plants. We can also measure the hydrogen isotopic composition of our leaf waxes. So both oxygen and hydrogen, and so you'll see this called delta 2H. Deuterium, or sometimes you'll see this also called delta D, because deuterium is the heavy isotope of hydrogen. And both oxygen and hydrogen will trace what's happening in terms of the global water cycle. And so when plants make their leaf waxes, they're going to be using local precipitation, and that will incorporate some information about what's happening in terms of the water cycle. So if we look at this map, this is the isotopic composition of mean annual precipitation. What we can see here is we go up to the high latitudes, you have very negative or more depleted values, relatively more enriched values in the tropics. When we look at the hydrogen isotopic composition of an individual plant wax, if we're up here in the high latitudes, temperature is going to be the largest factor that's going to control that. So it's used as a proxy for temperature in the higher latitudes. And if we're up here in the tropics, here the amount of precipitation is going to be the largest effect because temperature variability is relatively small. So plant wax, deuterium, or hydrogen isotopic values are used as a proxy for precipitation amount or rainfall amount in the tropics. So the goals of this study, we're going to use our plant leaf wax isotopes to investigate both the vegetation and rainfall history of Northwest Africa during the Pliocene and Pleistocene. And I'd like to point out for a second here all of the undergraduates that helped me with this research. So this is a project that I've done primarily with undergraduates over quite a few years. So Ben Uren, Stephen Lucas, Tom Barrasso, Victoria Fu, and Joanne Johnson generated the data I'm going to be showing here today. So the time interval that we are studying is the Pliocene and the Pleistocene, so the last 5 million years of Earth's history. On the top here is age, going from today, 0 million years ago, to 5 million years before the present. And at the top, this is the global benthic oxygen isotope stack, and this is sort of a good representation about what's happening in terms of global climate. So as we go from the Pliocene, which was a relatively warmer world, less ice volume, and about 2.7 million years ago, we have the intensification of northern hemispheric glaciations. So on this diagram, you'll see there's all these little wiggles. And what these wiggles are showing is Earth switching back and forth between a warm interglacial climate like today and a cold ice age kind of climate. So these interglacial cycles, they intensify in the Pleistocene, and overall it gets cooler with ice volume as we move towards the present. And in the Pleistocene, we have a main event called the Mid-Pleistocene Transition, or MPT. And what's interesting about the MPT is that prior to this time, if we look at how often these interglacial cycles are occurring, they're occurring about every 41,000 years. And then after this time, they occur every 100,000 years. And the cause of the switch is presently under a lot of debate. One reason I'm interested in studying the climate of North Africa is that it's really important for the evolution of our own species. So this figure here, which comes from Maslin et al. 2014, shows different species for our human ancestors and when they were present, and also where they lived. So for example, our own species Homo sapiens is relatively young, going back to about 235,000 years ago. And it's often thought that some of these key transitions and steps in human evolution had to do with changing climate globally. But these theories have been hard to test because in a lot of places there are a lack of climate records from the sites that human fossils have been found in. So one of the first people to study some records from North Africa. So in 1986, the ocean drilling went to these sites around the North Africa continent and drilled all these different sites here. And they're labeled by these different numbers. And Peter Domenical was one of the first person to study these records. So what you can see in this plot, age is going, today is at the top, going to about 4.5 million years ago at the bottom. And for each one of these sites, so over here first are these sites in West Africa, and there's a couple sites over here in East Africa, like this site 721, 722 is this record over here. And what Peter Domenical did is he studied the dust flux in all of these cores. And based on looking, and these are the records that you're seeing here. So this is just dust flux. So how much dust is he seeing in these ocean sediments? And here is the compilation of all these records. So based on studying the dust flux, Domenical, in two different papers in 1995 and 2004, concluded that over about the last 2.8 million years, North Africa has been getting more arid, and that step-like shifts in the climate happened at about 2.8, 1.7, and 1 million years ago. Now, these dust flux records, since they've been generated, have been the source of a lot of debate. And one of the reasons is that dust flux itself is sort of a complicated proxy. So, for example, just because it gets more and more arid, it doesn't mean that you're going to have more dust blowing off the continent. And some studies have shown that maximum dust flux will occur when you have like a landscape that's undergoing repeated like wet and dry cycles. You might get more dust flux then, rather than if it's just like totally arid. So in 2009, a study by Martin Trouth went back to these same dust flux records. They used the exact same data as Peter Domenical, but they just used a different type of statistical analysis, and they reached an opposite conclusion, that the African continent has not been getting more arid since about 3 million years ago. So one of the goals of our study is we wanted to use leaf wax hydrogen isotopes, which is a completely independent proxy. So it's a proxy for rainfall amount in the tropics, to determine has it been getting wetter in the last 3 million years. So that's one of our first goals of this project. The second is to look at changes in the vegetation. So there has been some prior work done looking at C3, C4 vegetation shifts on the African continent. And these figures you're seeing here come from a paper that was published in 2019 in Nature Geosciences by Pratibha Pallisar and co-authors. And what they did is they went to these different sites, so sites 659 and 959 in North Africa. And the site 659 here is located pretty close to the site I'm going to be talking about today. And they also looked at two sites in Eastern Africa, so sites 235 and 241. And they measured the carbon isotopic composition of leaf waxes in those records. And so those results are shown here. So here is age, and this is going from 0 to 17 million years ago. So they looked at multiple sites over a long time span, but at low resolution. So like the spacing between these individual data points is something like 500,000 years at a lot of these sites. So if you look at first, just like these orange circles here, this data comes from this ODP site 659. And basically what they found is that on both sides of the African continents, and here is our leaf wax carbon isotopic values. And so our scale is going from more depleted or more negative values. So this is showing C3 trees. And then on the right of the screen, when it plots on that side, it will indicate more inputs from C4 plants. And what they see is about 10 million years ago, you start to see the rise of C4 plants across the African continent. So in the older part of the record, we don't have C4 vegetation present on the African continent. It appears on both sides in both Eastern and Western Africa about 10 million years ago. And we can see that today we're focusing on the last 5 million years of the record. The trend in this last 5 million years is one towards gradually increasing inputs of C4 plants. Again, they looked at this in quite low resolution. So today we're going to take another look at the same time interval. So here they have about a spacing of about 500,000 years between their data points. In our study, we have a sample spacing of 18,000 years. So another result from that POLISTAR study, so this slide here, this figure on the left is the same plot we just saw from looking at their C3, C4 vegetation shifts. They also measured the hydrogen isotopic composition of their leaf waxes. And so with this proxy, if it was wetter, those values are going to be more depleted, plotting towards the left. And if it's arid, they're going to be plotting more towards the right. Now, if this gradual shift in the vegetation since like 10 million years ago, as we go to more C4 inputs, if this was driven by changes in the amount of precipitation, we would also expect that in this plot for the hydrogen isotopes that we would see a similar trend with it getting more arid. But that is actually not what we see. If we look at this record for the hydrogen isotopes, yeah, there's some trends in here, there's some variability, but there's not an overall trend towards the more arid end of this plot. So because of this, these authors reached a conclusion that this increasing C4 expansion in North Africa must have been driven by declining atmospheric CO2 concentrations rather than changes in rainfall. If we go to other studies from the eastern side of the African continent, we can see sort of similar things. So this is leaf waxes. These were also shown on the last slide in the POLISTAR study. This is carbon isotopes of elephant teeth, and this is of pedogenic soil carbonates, which also reflects changes in C3 and C4 plants. And in all these different proxies throughout East Africa, we can see that the onset of C3, C4 vegetation happens around 10 million years ago. And in the last 5 million years, there's also quite a bit of variability. One of these records from East Africa was done by Liddy et al. In 2016. And here they also looked at leaf wax carbon isotopes, which is the same technique I'm going to be looking at today. So these are their results, and that site comes from right here, Deep Sea Drilling Program Site 231. And this record spans from 2 to about 5.5 million years ago. And on this plot, more C4 vegetation is plotting up towards the top of your screen. So as we look at this record, we can see that in the interval from 5 to 2 million years ago, there's quite a bit of variability between shifts from C3 to C4 plants. And one of the other questions we had was, is the C3, C4 history of Eastern Africa the same as what we're going to see on the western side of Africa? And one of the reasons to think that it might be different is that there's been quite a bit of tectonic activity throughout the entire East African Rift Valley that can cause some local changes in the climate or rainfall. So we wouldn't necessarily expect to see the same shifts happening across the entire North African continent. So we're taking a look at some data from our own site. So we studied ODP site 660. It's located just to the south of this site 659, which is one of the sites that Peter Domenical studied with the dust flux. It comes from a water depth of over 4,000 meters. The core itself is 165 meters long, but for the study we're looking only at the uppermost 72 meters. And the study interval it covers spans from about 0.47 to 4.1 million years ago. And as I mentioned before, our samples are spaced about 18,500 years apart. Currently, the age model for this is based on shipboard measurements of biostratigraphy. So this is like first or last appearance species, data from different species, and the red data points are for paleomagnetic reversals. There is not yet an oxygen isotope stratigraphy on this core, but for the purpose of this study, where we're just looking at broad trends over the Pliocene and Pleistocene, this age model is perfectly sufficient. So the site receives dust from several different sources. So in June, July, August, the Saharan air layer or the African easterly jet will transport dust that's sourced like in here from the central North Africa offshore. And then in December, January, February, the site can also receive inputs that stem from Northern Africa. So the dust comes from these two sources offshore. Now, other studies have looked at the geochemical composition of the dust using radiogenic isotopes and have determined that the source areas for this dust has not changed in the Pliocene and Pleistocene, although it's thought that at times the strength of these winds has varied. Okay, so in the next series of slides, you're going to start to see our own results from ODP Site 660. So on the top here, I'm showing the global benthic hydrogen isotope stack just for reference. So this is like our global record of what's happening between the Pliocene and the Pleistocene. In our own data, you'll see in the light colors with the individual data points, this is the raw data points, and then the thicker line is like a 5-point running mean. So just like a smooth line to help draw your attention to the trends. One of the first things that stands out here is, now we're just looking at concentrations of leaf waxes. So how much leaf waxes per gram sediment are we getting at our site? And you can see that there's a big change that happens starting about 2.7 million years ago, which is coincident with the intensification of Northern Hemisphere glaciations. So prior to this time, we have lower concentrations of leaf waxes. After this time, it really jumps up and it gets a lot more variable. This result is in really good agreement with numerous other studies that have looked at global changes in primary productivity and also the strength of the trade winds. It's generally thought that the strength of the trade winds increased about 2.7 to 2.5 million years ago, and certainly our record seems to be supporting this. On the bottom here, these are alkaline mass accumulation rate records. This is also a proxy for global productivity, and you can just really see that it's low prior to this time and much higher after this time. So everything is pointing to increased trade wind strains after about 2.7 million years. We can now take a look at our carbon isotopes measured on our plant waxes, and this is the record we get here. So 0.5 to 4 million years, and in this plot, more C4 plants is plotting upwards. So as we look at this record, we can see that there's definitely a lot of variability here, and there's an overall trend from more C3 plants in the Pliocene to more C4 plants as we get into the Pleistocene. But you'll also notice that this is not a monotonous, continual gradual shift, right? There's a lot of variability here, and there's also some reversals. So there are a few times when we see reversals to more C3 plants. And for example, some of these I've highlighted here in green, happening at about 3.5, 2.7, and around 1 million years ago. Overall, there's some pollen data that overlap with this record from ODP Site 659 to the north. It doesn't cover the entire record, but where there is pollen data, it does agree well with the results we have here. So one question we had is we could see in our C4 plants, there's this overall trend here to more C4 plants as we go from the Pliocene into the Pleistocene. And we wanted to know, is this related to increasing aridity? And to answer this, we need to look now at our plant wax hydrogen isotope record. And this is the record that we can see here in the bottom. So these are our plant wax hydrogen isotopes. And if we have an increasing trend to more C4 inputs, we would also expect to see that down here, we would have a similar trend. But we don't see that actually. So kind of like the Polisar et al. Study, we see that there's variability in the leaf wax hydrogen isotopic record, but it doesn't seem to be mimicking the changes that are happening in the C3, C4 plants. And if we look at what's changes since the last 3 million years, there's not really an overall trend here. If I fit a trend line through here, it ends up being more or less a flat line. So this is not supporting the idea from Domenical that there's been a progressive aridification of the African continent since about 3 million years ago. Another interesting thing is that we see in the Pliocene, from 4 million years to 3 million years ago, it goes from quite a bit wetter to being, from being more arid to being wetter. And this is kind of surprising because the Pliocene is often considered like a really long period of warm, stable climates. And in fact, we're seeing that in North Africa, this time period was quite variable in terms of the climate. So another thing we wanted to do now is we wanted to compare what's happening between our carbon isotopes between Eastern and Western Africa. So here, this is our data from West Africa. This in pink is this record I showed at the beginning from Liddy et al. From the eastern side of the African continent. And at the bottom here, I've just plotted the smooth data from both sites so we can now compare. And these two records will overlap between about 4 and 2 million years ago. So where we have overlapping data between the two records, they are in quite good agreement with each other. So here we can see this down here, we can see the same patterns are mimicked in both records. And this is showing that basically the shifts between C3 and C4 plants seem to be occurring at the same time on both sides of the African continent. Another interesting thing from East Africa is this record from looking at pedogenic soil carbonates. And so pedogenic soil carbonates also are one of the main techniques for reconstructing past C3 and C4 plants. And here the authors have compiled a number of data sets that I think is something like 700 or 800 samples that span the entire East African rift system. And they've converted this value to a fraction of woody cover. So basically in this plot, more C4 plants are going to plot like up in this direction. So again, towards the top of the screen. So this study was done by Quinn and Lepre. And what they found is they have this overall excursion to more C4 plants throughout the East African Rift Valley, which is also what we see on the West African side of the continent. But one of the really interesting things they found is they have this excursion to more C3 plants that occurred during the mid-Pleistocene transition. And interestingly, we also see, this is when we also have quite a large excursion happening on the West African side of the continent. So we also see a big excursion to more C3 plants during the MPT. Now, the authors of this study suggest that the reason that the switch occurred was probably due to a short-term rise in atmospheric CO2 concentrations, which may have been accompanied by an increase in temperature. And they also pointed out in this study that just because this happened in East Africa, that maybe it was a local thing, and we wouldn't necessarily expect to see it on the West African side of the continent. But we do indeed now know that this transition happened across the entire North African continent, as we can see it on both sides. What is also really interesting about this time interval, so what we're seeing now is this is the same record here from 3 to 0 million years ago from Quinn and Lepre, and now it's just the axis here is flipped, and now it's focusing in on the time interval from 0.5 to 2.5 million years ago. So this is that same data from the pedogenic soil carbonates. More C4 plants is now plotting off to the right on your screen, and this is the mid-Pleistocene transition. And what's really interesting is what they note is that there's this species of early hominin, so Paranthropus boise, so this species here. And this was an interesting species because it had a diet that consisted mainly of C4 plants. So some species of early hominids were more flexible with their diets, and they could eat both C4 and C3 plants. And here the authors proposed that because this diet was so specialized and had an inflexible diet where it only ate C4 plants, that when we started to have this transition back to more C3 plants, that was the reason that this species went extinct. Okay, so I also had some data for sea surface temperature. I'm going to kind of go over this pretty quickly just because I'm running out of time here. But we also looked at sea surface temperature at the site, and one of the main ways we do this is looking at long-chain alkanones. So these are produced by coccoliths, and these organisms are found everywhere in the oceans. And the way that this index works is we look at the difference between those that have 2 or 3 double bonds in the structure. And what happens is as it gets warmer, you have less of this one that has 3 double bonds. And when it's colder, you have more of the one with 3 double bonds. So this is called the UK37 index. And we can use this as a way to reconstruct past temperature. So we wanted to know at our site, was this time interval of the mid-Pleistocene transition also a little bit warmer? And so overall, this is the record we get from ODP site 660. We get what we expect where the Pliocene is relatively warmer. As we go into the Pleistocene, we see this trend to more cooling and it gets more variable. Our record has a sample spacing of 18,500 years. Nearby at site ODP 662, Tim Herbert has generated this record that's much higher resolution. It has a sample spacing of 2,000 years. And overall, if we were to put these records on top of each other, they agree quite nicely. It's just that this Herbert et al. Record is in much higher resolution. And if we look at in our own record for the mid-Pleistocene transition about 1 million years ago, we see relatively higher sea surface temperature compared to like the time before and after it, but it doesn't stand out as being really unusual. And so what has also been noted from prior studies of this region of North Africa is that, in some studies have found that there's little relationships between sea surface temperature and aridity and dust in North Africa. And other studies have suggested that in this part of North Africa, the sea surface temperature gradient or changes in the tropical Atlantic thermocline depth are more important for controlling rainfall amount in North Africa rather than just the sea surface temperature. So these are some ideas that we're going to look at to investigate with this study in the future. And we are also looking at presently looking at membrane lipids produced by bacteria that are found in soils to reconstruct the temperature of the continent of the source area of the dust itself. So overall, we use compound-specific isotopes. The hydrogen isotope record does not support a progressive aridification in North Africa since about 3 million years ago. But we do see that over the past 4 million years, this region experienced an overall shift from more C3 plants, which are trees in the Pliocene, to increased C4 plants or grasses in the Pleistocene. This transition towards more C4 plants was interrupted by several periods of C3 expansion. One of those happened at about 1 million years ago, coincident with the mid-Pleistocene transition. We see several, between Western and Eastern Africa, we see similar patterns in our C3 and C4 shifts. And again, the mid-Pleistocene transition stands out as being pretty unusual on both parts of the African continents with that shift to more C3 plants. And overall, I hope I've shown you that we can use ocean cores to study our past continental climate. So with that, I would be happy to answer any questions you may have. Thank you.
Thank you, Isla.
Thank you.
Thank you. Okay, we have a question here by Christopher.
Hello.
Yeah, we can hear you.
Go ahead.
I was curious about what effect the solvents you use, like DCM, has on targets like organic microfossils, like spores or pollen. Is the effect of the solvent strong enough that it actually makes the fossil too brittle, or are you or other people still able to take a look at the fossils after you've used the solvent to extract the compounds?
Yeah, that's a great question. And I actually don't know for sure the answer to that, because usually what we do is when we work with people that are looking at the pollen, we will both take a little bit of sediments. So I'll take my own sediment sample for the organic analyses, and like the palynologist will take a separate sample for pollen or microfossils. I don't think the organic solvents are strong enough to affect the pollen, because as far as I know, to study pollen, you you need to use like hydrofluoric acid to isolate the pollen from other parts of the sediment. But what we do is in organic chemistry, like what we do with the sediment is after we freeze dry it, we will take a mortar and pestle and we'll homogenize it, which I think that would probably be the bad part for the pollen and the other microfossils. Did that answer your question?
Yes, thank you.
Thank you. We have another question by Nandi.
I had a couple of questions. The first one was when you showed the carbon and the hydrogen isotopes, I saw that it was the C31 that you specifically measured. So I've seen in different literature, some have measured C27, C29. So is there a difference between the carbon chain length that you choose in those isotopes?
Yeah, so the reason we measured the carbon isotopes and the hydrogen on the C31 N-alkane is because it's the most abundant. For a lot of the samples, we've also measured C27, C29, and C33. But it was just the C31 N-alkane is the most abundant. So throughout the entire record, we've been able to measure carbon and hydrogen on the most samples. Particularly the hydrogen isotopic measurement takes a lot of material, and I forgot to mention that before, but our hydrogen isotopic record is of slightly lower resolution just because not all samples yielded enough material for us to be able to make that analysis. But typically we measure, the most abundant one. And we also look at the trends. So if you look at if I was to plot the data for all those other chain lengths, it would give the same pattern. So it doesn't really matter what chain length we're looking at, we're getting the same patterns out.
That I have a follow-up question is that when we see that there's a change from C3 to C4 vegetation, there's also the increase in carbon chain lengths associated with it, right? Or is that idea wrong?
If we look at that one is a little more complicated. I'm trying to remember what the chain length distributions look like. There's not an overall, if we look, I could pull that up for you. I don't know if you're around after the talk to look at the chain length data, but the chain length data doesn't have there's some variability there, but it doesn't have the same overall trend. And I think one of the reasons for that is chain lengths are harder to study because in this area, there are some species that lived in the Pliocene that they're not existing today. So we don't really know in the past what some of these species look like.
Okay, and the one final question I had was when you showed that there was you're going to do the study on the sea surface temperature. I know that you already worked on the BIT index and the TEX86 index. So are those something that cannot be conducted on this score, or are these something you're looking to do in the future?
Yeah, we do have TEX86 data from this site. At this site, so TEX86 at some sites is a sea surface temperature, and at some places it's a shallow subsurface temperature. At this site, so in this area of Northwest Africa, there's been a lot more work done looking at TEX86 on more recent timescales, like a couple 100,000 years. And so what we get at this site is we have the alkanones or the sea surface temperature, and the TEX86 is more like a 500-meter depth temperature, so it's shallow subsurface. And actually there's a paper that was published in 2010 by Raquel Lopez dos Santos in Earth and Planetary Science Letters. And she uses the difference between the alkanones and TEX86 as a proxy for the strength of the Atlantic meridional overturning circulation. So we do have TEX86 data. I didn't show it today because I couldn't get through explaining that many proxies in a seminar, unless it would be several hours long. But yeah, we do have that data, and it's probably a shallow subsurface temperature.
Gotcha. Thank you for that. It was a great talk. And if I'd like to hang around after the talk a bit.
Oh, great.
Thank you, Nandi. We have another question by Catherine and then Jackson.
Hello. First of all, thank you so much for giving such a wonderful presentation. I thoroughly enjoyed it. I was just wondering if you'd be comfortable sharing any contact information so that way we might be able to reach out to you in the future.
Yeah, of course. So my email is isla, ISLA, at umass.edu.
Thank you.
I can also share the information with the students from the seminar, no problem.
Great, yeah, I'd be definitely happy to answer other questions via email.
Okay, now Jackson, Ryan.
All right, so my question is, if you guys were talking about hydrogen isotopes, how were they actually getting them if hydrogen is a gas, and where did they originally get it from, and how did they go about obtaining it?
Yeah, that's a great question. So the hydrogen comes from the water, so H2O. So basically when it rains, that plant is taking the local precipitation, and the plant roots uptake that. And then the isotopic composition of the plant waxes is going to be a reflection of the isotopic composition of the rainfall. And then there's a biosynthetic fractionation, so like an offset that happens within the plant leaf as it makes that leaf wax. But that there's quite a few studies that have studied that biosynthetic fractionation, and so we kind of just assume that it's the same back in time, and we don't really know, but it probably is a sort of reasonable assumption. But basically, the plant waxes are going to reflect the composition of the water that the plant's using. If you go into, there are some greenhouses, like let's say we had some greenhouse study, or we have our plants growing in a little growth chamber, and for some reason, like let's say we're watering them with something like distilled water, you can end up with some plants that have some wacky leaf wax isotopic compositions there.
So are you basically saying that distilled water would actually be really bad to water your plants with?
I think it is. I can't remember why, but I think there's some other reasons why you don't necessarily want to water plant, or maybe it's deionized water that's bad for the plants. But I know in laboratory settings, like if you're watering your plants with water that's not coming from rainfall, you can sometimes get some weird isotopic compositions in your leaf waxes.
Thank you.
Let me see if there's any other question. I have one. Do you have any question, Arnie?
Sure, go ahead, and I can ask a question.
Yeah, I have a question with the use of enrichment and depletion in Pelio studies. So usually people use enriched values as warmer, drier, and then deplete it as wetter periods, and sometimes that works really good, but and sometimes it doesn't. And so I'm wondering if you have been thinking about other atmospheric processes that may be changing the isotopic composition. So, for example, I use the information from hurricanes in Central America and the Gulf of Mexico, and so hurricanes can produce a lot of rainfall, but it's actually enriched in many cases. It's not really depleted. So it goes against the precipitation amount effect. So have you ever think about those kind of atmospheric processes, especially because I'm aware that in Eastern Africa, there is an isotopic anomaly